Hi everyone. As many of you know, I'm Ejе. My daughter is Emily, and I've set up the Kids with SPG3A platform together with Carter's family. And as promised, we have a new update, and we're lucky enough to have Dr. Joe Avellone and Dr. Blackstone with us today. So I'll hand them over to share the update with us.
Thank you very much, Ajay. And once again, thank you for your dedication to SPG3A, you and Juan. I'm here today with Dr. Craig Blackstone. He's our senior scientific advisor. As you know, he's a full professor at Harvard Medical School and head of a division of neurology, motor divisions, and actually many more things. But we're so pleased to have him guide our research. And I am Joe Avellone. I am the president of the Carter Foundation. What we'd like to do today is do our biannual update on the activities of the Carter Foundation, which are really focused on SPG3 and to some degree other similar kinds of hereditary spastic paraplegias. So if we could go to the next slide, please. I thought I'd take a step back as we get started to give a little context of what we're doing. A lot of the activities, and in fact, all of the activities that we've been undertaking for the last several years have been in the world of what's called translational medicine. And that is taking from the understanding of the basic biochemistry, which have been worked out over many years, much of which in Dr. Blackstone's laboratory at NIH and other places, and trying to take that and learn from that in an animal model or several animal models in order to prepare for the potential for future therapies. And the reason the animal models are so important is they are closer to being a human. And so we use a variety of animal models to prove out potential therapies. And we have been evaluating three, I say four here because we divided the drugs into two, but three potential therapeutic approaches to SPG3A. And once again, these hypotheses have been worked out in Dr. Blackstone's laboratories, both at NIH and now at MGH. So the three living translational medicine models we use are shown on the slide. The first is using human cells in a petri dish. So we've been able to take, and now people can take, stem cell-like cells and promote them into actual human neurons that grow in vitro, so in a petri dish, and then promote them into the exact mutations that we're studying. So that is what has happened. And a lot of that work is being done by Dr. June Lee at the University of Illinois Medical School in Chicago. We also have been using fruit flies as a model, and you may ask why. Well, fruit flies are very good in studying genetic type diseases because they replicate very, very rapidly. So you can see generations of fruit flies at 3 or 4 weeks. And that's being carried out by a colleague of Dr. Blackstone's at the University College in Dublin. And finally, we have been developing a human mouse model, a human mutation mouse model. So a mouse model at the Jackson Laboratories, which has been changed to have Carter's exact human mutation in the mouse. I know that sounds strange, but that is exactly what it is. The DNA that the mouse has is mouse DNA, except for a little piece that has been inserted, which is Carter's mutation, which is an SPG3A mutation. And so with those three translational medicine models, we've been trying to evaluate several approaches. And the first would be a drug approach, because that, of course, would be easiest to administer and would be also, in many cases, often have a safety profile that we already know about, because many of these drugs have been used or at least tested in humans before. And then the second area is to use one of the more modern cell and gene therapy approaches. In this case, it's called ASO, and we'll talk about it more later. But it is to take essentially silence a part of the gene that may be causing some problems. And the third, and probably most advanced, is to study the ability to actually edit the gene, actually edit a gene in our patient, that it then has the correct sequence that would then make the correct amount of elastin-1, which is the protein is missing. So just as a backdrop, that's what the endeavor has been for the last several years. And we've been making some progress, which I'm happy to report today. But usually in the way that science always is, which it doesn't go in a straight line. So we have been learning some important things. If we go to the next slide. first of all, speaking to the iPSC project, so that is the human neuron with Carter, in this case, Carter's mutation, which is an SPG3A mutation in a petri dish. Um, we did have been studying several drugs. And well, Craig, maybe I could ask you to kind of describe where we are with the iPSC studies.
Yeah, sure, Joe. Thank you for including me, and thank you, AJ, as well for hosting this. As Joe has mentioned, we are doing several studies in iPSCs, and what these are cells that are taken from a patient, skin cells, and you can actually reprogram them to stem cells and then use those stem cells to make nerve cells. So it's a way to make a human nerve model from actual patients. And of course, we can control these studies by fixing the mutation or using cells from patients who don't have any neurologic diseases. And because these cells are made into neurons that are similar to the ones that cause the disease, we use that as sort of a model, as Joe said, for to see if we can get an improvement in the cell model. And then, of course, we move on, as Joe also said, to in vivo models or animal models, which again get a little bit closer to people instead of just being a cell line. Now, the first study we completed, we did several studies with at June Lee's lab, and we did see that there was some benefit in neuronal health. And of course, when we say neuronal health, we mean things like axon abnormalities or varicosities or things that we can see that aren't normally present in normal cells. And what's important to know is even if we reverse these, we're not sure that these are relevant for the human disease, and that's why we have to use multiple model systems. However, it did have benefit, which was hopeful in the human iPSC line. Of course, when we see that, then we think, okay, we want to check that in other models as well.
We also are testing some other drugs that are a little more complicated to describe, but they all have to do with neuronal health, as Dr. Blackstone said. And one of which that also starts to show some early activity, at least so far in the petri dish, is the so-called BMP signaling pathway. It's just another complicated biochemical pathway in the neuron, but it does show with drugs that can inhibit it that some positive activity. Once again, these are very preliminary, and we are planning to study several more drugs. You go to the next slide, please. I mentioned that we were doing some studies in the fruit flies, and I think we learned something pretty important in the fruit flies around SPG3. And so they studied a mutation, which is not the exact human mutation, but a similar mutation in the fruit fly, which has a neuron that goes down its wing that is easily or more easily observable than many other kinds of primitive animal models. And we did show that there was a difference in the benefit of trying to use, in this case, the lipid modulator again, with one version of the genetic mutation, that is the version that was a de novo mutation versus the other, which was an inherited mutation. And so that raised a very important and seemed to demonstrate a very important potential concept that is leading us in a direction in our research going forward. It raised the possibility that kids that are born with what's called a de novo or a spontaneous mutation show a lot of symptoms and their disease quite early compared to kids that inherit it from one or the other of their parents in a normal genetic way. And that is pretty important because it may be a different mechanism. It may be that the kids that actually start out in life feeling pretty good and acting pretty good and only later develop the disease, which is the common pathway for people who inherited it, are different from kids who have it right out of at birth basically and struggle with it. And it may be that there's without going into too great a detail, it may be that there's a mechanism in which the protein that's missing is elastin-1. And in the case where kids are born with the disease so dramatic early, it may be that it's not that they're missing one of the DNA expressions of elastin-1. It may be that there's a bad version of it that's inhibiting the other version of it, and that's why their symptoms are so bad. Well, that is, I think, it sets up the possibility of a potential treatment that we are pursuing, which we'll describe in a little bit. So even though this Drosophila project didn't show anything exactly the way we wanted it, we learned something pretty important about potential different mechanism that we are pursuing with our other projects. Craig, would you wanted to comment on that at all?
Yeah, I would emphasize, thanks, Joe. That was a great description. And as you said, one of the things that is important for us to do is to understand why some of the children with de novo mutations get a much more severe presentation. And as you outlined very nicely, the majority of patients, let's say, that have a typical presentation might still have the abnormal protein produced, but it might just be inhibiting the elastin protein function in some way. However, as you described in the flies in particular, these particular mutations, as Carter and other children with these more severe forms exhibit, they might have something additional. There might be perhaps in addition to the other function or the other dysfunction, an additional new gain of function that's toxic. And as you mentioned correctly, one reason that we might not be able to get medications to work for this is that the important thing we need to do is to sort of delete or remove the abnormal protein. And that's, as you said, that's why we start to think about, okay, is there a way we can use new technologies to remove the bad protein? And one thing I could mention that in other patients with this disorder and in other families, we do have indications that it's safe to remove one form. And since all patients with HSP3A, or the vast majority of them to be exact, just have one allele affected. In other words, they have a normal gene too. So if we can remove the abnormal gene, they still have a normal one that should be sufficient to perform all the normal functions of this protein in these patients.
Yep. Very good. Thank you. So if we go to the next slide. Can we go to the next slide, please?
Oh, did it not work? It's here.
Nope, I did and then came back. So go ahead one more. Oh, you know what? I'm sorry. I'm sorry. That was the next slide. My fault. We are doing some other things with the Drosophila project as well. So, and that is we're studying some of the drugs that we're also studying in the Petri dish with the neuron. Once again, an animal model, it's a little farther along. And so between the Petri dish, neuron, and these Drosophila projects, I think we'll have a pretty good review of drugs that seem to be active in helping, at least helping in these neurodegenerative diseases. And once again, as we get down to more definitive cures, as Dr. Blackstone has often said to me, it may be a combination of things that actually optimize the treatment. So it's worth trying to understand these drugs, even as we're looking at some of these more advanced therapies as well. If you go to the next slide. I'm sorry, yes, there we go. So based on learning that the de novo patients that have the mutation at birth and as a spontaneous mutation may have a toxic kind of elastin that's being done, there is the possibility of silencing that bad gene, as Dr. Blackstone was saying. And there is an effort underway to try and test that theory. There is a company in San Diego, California, known as Ionis, and they have actually been pioneering some of these so-called ASO therapies. And they have founded a foundation along the way, which takes on some of these very rare versions of these hereditary spastic paraplegias or other kinds of rare neurodegenerative diseases. It's called the n-Lorem Foundation. And so the Carter Foundation reached out to them, and Carter, the patient, has been accepted to test out a potential treatment, an ASO treatment, if they can develop it. And so we're now in the process, and the process would take over a year to a year and a half from here even, to go ahead and design the exact sequence of a chemical that could, once again, intercept the bad gene's output and silence it so it doesn't get made into a bad kind of elastin, allowing the good elastin to serve the purpose. This is a trial of one. This is not the way clinical medicine would typically be developed, where you'd have trials with many patients. But given the circumstances, what they can do is at least using a patient, if we can make it safe enough and good enough, to test whether this works before it gets expanded to use in other patients. So once again, if you go to the next slide. we are in this process, and we are in the second arrow from the left, which is the discovery process. So right now they are actually taking Carter's exact genetic mutation and testing various sequences of chemicals that would have the property of intercepting the bad output from one of the DNA strands and preventing it from making a bad version of the protein. From that, and that takes many, many, many months in order to find the exact sequence that they think will be optimal and safest. And then it has to be tested for safety in a mouse model and in some other ways before it ever gets to the point of potentially having it used in the trial with Carter in this case. Craig, is there anything you would like to add to this?
Yeah, Joe, that was a great description. One other thing to note, though, is that as n-Lorem is doing this, looking for different potential sequences, they can also take advantage in humans or what are known as polymorphisms. And those are
Right.
just changes that occur that don't cause disease, but that might be present in a large portion of the population. But they still allow you to silence the gene. And I know that's complicated, but the advantage of that approach, and that's one that they will be testing, is that the exact siRNAs or antisense oligonucleotides that are used for Carter could potentially be used for other children with other mutations,
Yeah.
that it might have much more broader applicability than just one patient. So our hope is, as you said, is to go from n equals 1 to n equals a few or n equals several next. And we might have the opportunity to use exactly the same sequence or the
Sequence.
same tools that they generate. Now, that may not be the case, but it is important that we will be able to move, I think, if this is safe in Carter in the initial test, to move this approach onto other children as well, hopefully relatively quickly.
Yes, that's an extremely important point. Um, okay, we go to the next slide, please. So the third category of models that we've been using is the so-called mouse model, and Jack in testing and experimenting, doing various kinds of studies in very complexly developed mouse models. And in this case, they have taken the exact mutation of this SPG3A variant of Carter's and grafted it into the DNA of the mouse. So it's a human mutant mouse model. So the first trial that we ran in it last year was to test out that lipid modulator that we talked about up front. And as we sometimes find, it did not show the benefit that we had hoped for in this human mutant mouse model. So even though it worked in the Petri dish, and there might have been some evidence that it worked in some parts of the fruit fly population, it did not work in the mouse. So the next step is to try and understand what did happen in the mouse. So the mouse experiments have come to an end, and there is now the effort going on to understand both what happened in the brain and also in the neurons, and also from studying in the blood other kinds of biomarkers to see what kind of activity and things we can learn. So I think where we are with this in the Carter Foundation approach is that I'm sure will result in a paper whose methodology and also the learnings will be important for research generally around SPG3A. And it may lead to some other things that we may find as we've talked about before. I think we're going to go ahead with a paper here in the Carter Foundation and probably not pursue it in our set of projects, but it may play into other projects by other researchers going forward. If you go to the next slide. So then the next thing we want to do with the mouse model is to do the third or fourth of our hypotheses, which is the actual gene editing itself. So now you've probably heard of CRISPR, Cas9, or the idea of gene editing, and that's what this is. This would take the exact mutation and with a very precise sequence that was developed called base editing, it could actually change the DNA, in this case, Carter's DNA's mutation, and hopefully back to normal. So we are going to do that. Of course, if that worked in a mouse, then that would be a very important discovery for the potential for what likely would be a cure. in humans. Don't want to overpromise that because we haven't gotten to the mouse yet, but we are starting that study. It is being designed right now, and our hope is to run this in 2026. The output would not be available for most of a year. Once again, mice take a long time to replicate, and then it takes a while for the tool, the base editing biochemical tool to work. So all of this would probably take through most of next year, but that is our plan. It would be very exciting if that worked out, certainly as exciting as the ASO experiment that we're doing. Dr. Blackstone, would you like to add to this?
Yeah, I'd just like to emphasize one point, Joe, about the mice too, is that the mouse is not an exact model of the human disease because it has a mutation in a second gene as well. And that's one reason we use other approaches, such as the flies and also the iPSCs, the neurons that we generate from patient cells. And the reason we had to do that in the mice was that the phenotype was relatively mild. And remember that when you think about the human disease, the nerves that are affected can be very long, up to a meter long in an adult. And of course, a mouse is a very small animal. So in order to get a good phenotype, which means like symptoms in the mouse that we can see if they get better, we had to mutate a second gene that interacts with the first one. And I know that's complicated, but it makes it so that the mouse is not an exact model of the human disease, but it has several of the same symptoms. And that could be a reason that some drugs don't work. However, for the editing approach, it will almost certainly work because we know if we fix one of those genes, that when only one gene is mutated of the two, the mouse's symptoms are very, very mild. So we will definitely see an improvement where the mouse with two mutations or two gene mutations is very severely affected. If we can fix one of those genes, the mouse will be relatively normal. And that's the reason we're doing this approach. But it is important to emphasize that all of these are models. Only the children have HSP. All these are model systems, and that's why we cut across several different ones because we can learn different things from each model system. But the mouse is a particularly good model system for the gene editing because, as you said earlier, Joe, that section of the gene in the mouse has been replaced with the human sequence. So any gene editing tools that we use in the mouse, in theory, should definitely work in human cells as well.
Thank you, Dr. Blackstone. If we could go to the next slide. So just wanted to recap what we said today. And the first thing I would say before even doing that is, especially with these more advanced therapies, but I should also say even with the drug therapies, if we find activity in something as in one of the SPG and one of the hereditary spastic paraplegias like SPG3A, some of these things could be transferable and useful, very useful in other forms of HSP. So even though our focus has been on SPG3A, depending on the exact findings, many of these things, whether it's the drugs or the ASO approach or even the gene editing approach, could be extrapolated and used in others. In the case of the ASO, as Craig said, if the so-called polymorphism would work out, that could be immediately transferable. In the case of the others, they could be developed using that kind of treatment platform. So I think this, all of these experiments have widespread potential importance, and we're kind of pleased that we're making some progress as we make our way through them. So today we just talked about the fact that we're using translational medicine models, that is, we're taking things that we learn in biochemistry effectively and move them in microbiology and move them into living models that gets us closer to how things might work in the human. And we've tested several things. We looked at the lipid modulator. We did not find that it worked in the mouse, even though it did work in the neuron. And so we're going to write a paper about that, which will explain all that and provide some learnings for other researchers. And we've also looked at testing a variety of other drugs, both in the iPSC model and potentially in the Drosophila model. And finally, or not finally, but the next thing is that we described that we are engaged with n-Lorem, a foundation on the West Coast that does cell and gene therapy treatments for very, very rare diseases. And they've taken on this one, SPG3A, for a potential trial of one human, which would be in about a year or a year and a half. But once again, could be extrapolated beyond that once we if we find something effective. And then the final is, in a sense, a little bit more futuristic, but it is using very precise gene editing, so-called base editing, um, in the mouse model to try and see if a gene editing approach is useful in this disease, which it very well might be. But once again, we need to test it in the mouse first. So I want to thank everybody for their attention today and their continued interest in the research of the Carter Foundation, and certainly in the science that Dr. Blackstone in large measure has developed. And we are thrilled to have him to continue to work with us. Any final words, Dr. Blackstone?
Yeah, thanks, Joe and AJ. I mean, again, I think that was a spectacular description. I think that you emphasized several key points. And one is that we have to use multiple different model systems and we have to try multiple different types of therapies because we never know which ones will work, which ones will be safe. But by trying several different ones in different model systems, we get, I think, the most complete picture of what will be effective for children with this disorder. And as I think you also emphasized, we may have to use things in combination. But that's not to, there's perhaps that one treatment won't be complete, but if we add it to another, we'll get that sort of additional benefit. So I think that we want to continue to pursue these different treatment approaches, and I do believe that ultimately the solution to this will be a combination of multiple ones, whether it's an oral pill plus a gene therapy, and that there's really great hope, as you said during your talk, that one of the things that's great about what the Carter Foundation is doing and where the field is now is it's really translational. All the research you're describing is toward cures. We understand a lot about what these genes do. Now we're at that stage in the science where we can emphasize, what can we do to treat these children? Not just what we can do to understand how these genes work, but what are the treatments that we can develop? And I think that's what's so exciting about this time is that, as you mentioned, we're going to start getting those answers very quickly.
Thank you. So once again, thank you, Dr. Blackstone, and thank you so much, Ache and Juan, for your diligence and work to have the website that we all refer to, and we're very pleased to continue to work with you.
Thank you so much for keeping us and the families on the platform in the loop. It's hugely beneficial for everyone to see the progress. So everybody's watching it very excitedly to see what's going to happen next. Thank you so much.
Thank you.
Take care, everyone.